Hello, everyone, and welcome to this webinar entitled A Deep Dive into Motor Unit Decomposition. My name is Colette, and I will be your host for today's webinar. In this webinar, we will have two speakers who I would like to introduce. Our first speaker is Dr. Julien Rossato. And Julien, he is a researcher currently doing a postdoctorate at the IRCCS Foundation Santa Lucia in Rome. His research focuses on the understanding of the control of movement and force generation, with the aim of restoring or augmenting them through innovative human-machine interfaces. Our second speaker is Joost Herijgers. He is an application engineer at TMSi who focuses on product management and software development. His work centers on enhancing user experiences and optimizing product functionality. Outside of his work, Joost enjoys riding his race bike, reading, and crocheting. Joost holds a master's degree in biomedical engineering from the University of Twente. Before I give the first word to our first speaker, Julien, I would like to go over some house rules. As you can see, your microphones and cameras are automatically muted and turned off. So if you have a question during the webinar, please write your question in the chat box. After the presentation and demonstration, I will go through the questions one by one. Again, I would like to welcome you all and thank you for being here. I hope you all enjoy the webinar, and we will now start with the presentation of Julien.
So, hello everybody. So first, I would like to thank TMSi to give me this opportunity to present one of my work. And today, I would like to introduce you to an open access tool that allow you to identify motor unit activity in real time. So first, I would like to begin with the object at the center of this work, the motor unit. So composed of an alpha motor neuron and the muscle fiber it innervates. It is a basic unit of musculoskeletal system, and it is the smallest control element that the central nervous system uses to produce force and movement. So we have between 10 and several thousand motor units per muscle, and the human body is composed of more than 600 muscle. So the musculoskeletal system is extremely redundant in the sense that multiple control solutions can achieve the same movement result. So this allows a certain flexibility in the production of movement. So to understand the control of motor units, many studies have looked at the activity of motor units. So here you can see the activity of two motor units, 1 and 2. And for each vertical line, you have a discharge from the motor unit in question. And from this activity, it is possible to obtain the discharge rate of these two motor units during the force ramp. So we can see that this motor unit behave very similarly. So not just to follow the progression of the force, but also during the force plateau. And so this observation have been made in many muscle and has led to the conclusion that this motor unit must receive the same synaptic inputs to behave in the same way. And so this is referred as a common drive. However, recent studies have highlighted contradictory results. For example, Formento and colleague studied the behavior of motor unit within the bicep brachii during different tasks. Here we can see the behavior of two motor units during an isometric supination. And so motor unit number 2 is recruited earlier and has a higher discharge rate. However, if we look at different tasks, we can see that the participant is able to recruit specifically either the motor unit 1 or either the motor unit 2. And so the size principle is no longer respected. And so these participants show a remarkably flexibility in controlling these two motor units, suggesting a specific command for motor unit 1 and 2. And so to go further in understanding the control of motor unit and to identify their discharge pattern in real time, could offer a lot of augmentation or restoration potentials. For example, here we can see a person controlling a cursor. So the cursor is here, and is controlling this cursor using the activity of 3 motor units. The motor unit blue is able to move the cursor upward. The motor unit red is to move the cursor bottom right, and the motor unit yellow is to move the cursor bottom left. And so this person is then able to write sentences by moving the cursor and simultaneously activating the 3 motor units to validate the input. It is also necessary to identify the activity of a larger number of motor units to better understand their control. So the reference technique for identifying motor unit is the use of intramuscular EMG, but as well as being invasive, it only allows the identification of a small number of motor units, 3 to 8 motor units, and during low intensity contraction. So an alternative method is the decomposition of the EMG signal into motor unit activity. So as you can see, the EMG signal is the sum of the discharge pattern of all the motor units. And so it is then possible to reverse the generative model of the EMG signal to find the sources of this signal, the sources, which is the activity of the motor unit. So to do this, it is necessary to increase the number of observations and therefore to use high-density electrode, either surface electrode or high-density intramuscular EMG. So the aim of my presentation is to present you iSpin, an open source software that I developed with Simon Avrillon. And our software is made to display to the participant a real-time visual feedback based on the activity of individual motor unit. So this work is currently a review preprint at eLife, so you can find it. And so to introduce you iSpin, I will start by describing the algorithm used to decompose the EMG signal. Then I will discuss an important step, the manual editing of the result. And finally, I will present the performance of real-time EMG signal decomposition by our software. So first, the decomposition algorithm. So it works in several stages. So first, the signal undergoes an extension to increase the efficiency of the software, and a whitening, which is a filter reducing the autocorrelation matrix of the signal. Then we use a fixed point algorithm to extract an independent source from the signal. So this algorithm iteratively apply a contrast function to the signal, and the result is a separation vector here. And this separation vector can be applied to the signal to extract sources here. And so the local maximum of these sources will then be considered as discharge instant. Estimation of these sources will then be refined using the fact that the discharge frequency of the source, which is the motor unit, should be stable during the force plateau. And so finally, the quality of the extracted source will be calculated in order to decide whether it is or not an activity of motor unit. So two values can be used, one of which is the silhouette value, the SIL. And so this silhouette value is a normalized value between 0 and 1. And this value is according to the distance between the centroid of the local maximum considered as potential and local maximum considered as noise. Here you can see example of the optimization of one separation vector and the sources associated during the fixed endpoint algorithm. And so separation vectors is iteratively optimized to maximize the sparseness of the motor unit pulse train. And to identify the spike from the noise, we use K-mean classification. The K-mean classification is applied to the local maximum. And with this, we can separate high peaks from the low peaks, and high peaks will be considered as spike, while the low peaks will be considered either as noise or as spike from other motor units. We validate this algorithm with synthetic electromyographic signal. So you can see here a raster plot of active motor units during a simulated trapezoidal contraction. So either at 10% of MVC, 20% of MVC, 30% of MVC, or during a sinusoidal contraction with the force varying between 15 and 25% of MVC. So each line here is one motor unit, and a spike is represented with a vertical line. And so the motor units in red are the motor units recognized with, identified with intramuscular high density EMG. The motor units in blue are the motor units identified with surface high density EMG, and in green with both intramuscular and surface high density EMG. So to measure the performance, we calculate the rate of agreement, which is the number of true positive divided by the sum of true positive, false positive, and false negative. And as you can see here, we had a very good result with a rate of agreement at 99% in average. So with experimental signal now, we obtain the activity of several dozen motor units among those that are active. And so in this example, we have 29 motor units identified from the vastus lateralis muscle following a force ramp. So as a reminder, with 3 of 8 motor units for classic intramuscular EMG, we obtain 3 to 10 times more motor units with this decomposition technique. However, with experimental signal, these approaches require some degree of manual editing of the motor unit spike train to check for false positive or false negative. Oh, excuse me. So manual editing improved the accuracy of the spike train. And so to do the manual editing, we use different information. So here you have the sources with the spike considered as action potential. And here you have the instantaneous discharge rate of each action potential. This is the black circle. And in red, you have the force production. And so manual editing involves several steps. First, you can remove erroneous firing rates. So for example, all these points can be removed. And you can also add discharge times that are clearly distinguishable from the nose. For example, here you have 2 local maximum, which is very different from the nose level. And so each time you do a modification of the discharge pattern, you recalculate the separation vector with the new collection of action potential selected, and then you reapply the separation vector on the EMG signal. And so you repeat this process until no outlier are present and all clearly distinguishable spikes have been selected. So here you can observe the effect of the manual editing on the result of a baseline contraction. So this graph represents the silhouette value, which is, as I remember, an estimation of the quality of the motor unit, and the variability of the instantaneous discharge rate for each motor unit. So you have a red point for the motor unit before addition and a green point for the motor unit after addition, and the 2 points are linked with a gray segment. And so if we look at the silhouette value, you can see that the manual editing improve this value. So the motor unit is therefore better defined, allowing us to obtain a better separation vector to apply during real-time tasks. And if we look at the stability of the discharge frequency, so the variability of instantaneous discharge rate, you can see that is greatly also improved by the correction of false positive and false negative, allowing us to obtain a better classifier to apply during real-time task. And so the result was similar for each muscle tested. So we test the vastus lateralis, the vastus medialis, the gastrocnemius lateralis, the gastrocnemius medialis, and the tibialis anterior. And it shows you the importance of manual editing before the online decomposition. So then, after performing a first decomposition on a contraction known as baseline or training contraction, we obtain for each motor unit a separation vector and an estimated sources from the signal. And so from these sources, we can have a classifier to classify the local maximum of these sources as discharge or noise. And so for real-time identification, we need this matrix containing all the identified separation vector and each classifier. During a real-time task, we then apply the separation matrix to the EMG signal. So we obtain a source function for each motor unit, and we can identify the local maximal in this function. And so we will use the classifier to categorize each maxima and identify the instant of discharge. It is then possible to provide online visual feedback on the activity of the motor unit identified. So to estimate the performance of the software, we use the same synthetic EMG signal by applying separation vector and classifier we obtain with the ramp at 20% of MVC, and we will apply this separation vector and classifier to the signal at 10, 30, or the sinusoidal around 20%. So first, we can look at the rate of agreement of the result when the motor unit filter were applied to the sinusoidal contraction here. So the rate of agreement was in average at 96%. We can look also at the rate of agreement, the sensitivity, the precision, and the false negative rate when we apply the motor unit filter to the simulated contraction at 10 or 20%. And so if you look for the result at 10, you can see that the rate of agreement remain high, and in average, it was 99%. On the contrary, if you look at the rate of agreement for the signal at 30%, you can see a decrease in the rate of agreement, which is at in average at 98%. And so it explained by a drop in the precision, which means that there is a lot of false positive. And we can explain that because by increasing the percentage of MVC, we have a new motor unit, and this motor unit can be merged into the motor unit targeted. For example, here you have the red dot represent the discharge time of this merged motor unit, while the green dot represent the discharge time of the targeted motor unit. And so this merge was caused because the recruitment of a motor unit sharing the same space as the targeted motor unit was turning into the merge of these two motor units by the model in the same pulse train. We tested the accuracy of the online decomposition with experimental signal as well in different muscle, and so the highest rate of agreement was with the tibialis anterior with 93% in average, and the lower rate of agreement was observed for the vastus medialis with 75% in average. We also estimate the accuracy of the real-time biofeedback with root mean square error between the real-time biofeedback and the corrected version with manual edition. So the root mean square error was in average consistently below 2 pulses per second for all the muscle. And so to be more concrete in practice, you can see here the discharge pattern of one motor unit during the real-time decomposition or after manual edition of this real-time decomposition. And you can see that the algorithm is able to identify period of activity than a period of rest, and is also able to identify changes in the frequency over time. Here you can see, for example, where the position of a cursor is a function of a discharge pattern of two motor units, one in a y-axis and another in one in the x-axis and another in the y-axis. And so even if we adopt the error produced by the real-time identification of two motor units, for each example, the position of the cursor in real time is very similar to the position of the cursor for the corrected version of this real time. So in conclusion, I presented iSpin, a freely available software that allows an EMG signal to be decomposed into motor unit activity in real time. And in addition, the algorithm performance has been measured, enabling accurate real-time identification. So the software compatible with Saga from TMSi is available in a public repository on my GitHub account. So here you can see all the contributors to this work, and thank you for your attention.
And then that's where I'll take over this presentation to actually demonstrate iSpin to all of you. So what we did is that we created a short video recording in which we want to talk you through and show the different functionalities of iSpin. So let's get started on that. So first of all, iSpin is a MATLAB app, and if you open iSpin, you get into this window. So in the top right, you can actually see that we already connected Saga, and on the top left, you can see that we, for instance, set the muscle name, which is in our experiment tibialis. Then next to that, we have set the various other settings for Saga, and you can see them being highlighted right now on the screen. So in this case, as said, the muscle has been specified, the sampling rate can be set, the amount of channels per grid can be specified, and on which channel of the Saga's auxiliary port the force sensor is connected. So then next up, we can actually start some visualizations to see whether our EMG signals are of high enough quality to be used later on. So in this part, the subject should rest as much as possible, and after doing that for 15 seconds, the following results are shown. And actually within this graph, it's mainly important to look at the amplitude of the bars that you can see. The amplitude can be seen here in the top left of the window. And in this case, we have an amplitude of 3.5 times 10 to the minus 3 millivolts, meaning that we only have a noise level of 3.5 microvolts, which is really good. What you will mainly see if there's bad channels is that one or two bars will really stand out from the rest. And then you know that you got to do something about this. And this is actually done in the next step because there's another visualization step in advance where the subject should also do some contractions. So you can see if there's any other effect happening to the EMG signal that's not desired. Because of course it's important that there are clean signals being used to decompose. So at the end of this noise check, which is a 30-second various contractions that the subject does voluntarily, we get these graphs. So you can actually see multiple things here. So there's 4 different columns that are highlighted, and the 4 columns all relate to a column on the EMG grid. And then there's, for each column, it specifies 1 row of the EMG signals. So as we recorded from a 32 channel grid, you can see 32 lines of EMG signals. There's a few things to note in this specific figure. So if we, for instance, look at this part, we see a clear resting period, followed by a voluntary contraction, followed by another rest period and a voluntary contraction. And actually after the last contraction, you can already see some behavior they think maybe there's something wrong on this. But actually this was our subject that could not really get his foot again in a nice resting position. And that's why you see some remaining EMG activity. After having these checks and evaluations, you can actually say if there's any signal that you would want to discard from the decomposition set. And for that, this dialog that's now on the screen can be filled out. We came to the conclusion that all of our signals were of high enough quality, also based on the previous noise check, so we accept them and don't discard any channels here. So that brings us to the next step, and maybe you've spotted it, but the panes in the top left have switched from training or from recording to training. And in this phase, we actually gonna train the record the training data for the decomposition. So we start off with a maximum voluntary contraction to normalize the force generated by the or recorded by the force sensor. And we do so by making some edits to the settings. So we specify the duration of this maximum voluntary contractions, which we chose to be 3 seconds. Then a rest period of 30 seconds, which is on the short side. And then finally, we record the offset from the load cell so that we can actually bring the baseline value to 0. Because of course, there's some force being measured because there's a foot resting on this sensor. Then after making those settings, we can actually start the MVC. So you can see that the button is being clicked and then the lever starts moving a bit. At this point, there's a resting state going on. And then as soon as the button turns green, then the subject should push as hard as possible or pull as hard as possible in this case. And then after completing that, he should return to the fully resting state, which again was a bit challenging, which is why you see the lever hanging around 3 right now. After performing the final MVC, because there's multiple repetitions, we can set the training parameters. This is done in the bottom left. So you can see that we're filling out the MVC target level, on which we're going to train data, and the duration of the plateau. And then a preview of the force profile is shown here. So you will see some edits happening to that as we fill out the values. Then the final thing that we need to set is the decomposition parameters. So there's the silhouette threshold and the covariance threshold. We leave the silhouette and covariance threshold as is, and we change the number of iterations to 75. Just for the purpose of giving the algorithm more results. Then we get to the actual phase where we're going to record data, and you'll be seeing a few things in this graph. So we already see the force profile on the right side of the screen and an actively recorded force starting on the left side. Our subject has resting his foot on a foot pedal, and now he will start to lift his foot and actually contract using the tibialis anterior. And here it's important that the force profile is matched accurately, because this is the way in which we can actually control the contraction intensity and thus control what type of recording we're doing and thus training the algorithm on. So actually you can see that it's quite hard to keep it exactly at this force level, but the subject does a quite good job on this. And then as we get towards the falling period, it again becomes a bit harder to follow this nicely, which you will see in the end results. But yeah, it's always a bit tricky to get this right. Then we're skipping ahead a bit, and we're doing so because now the decomposition is running, and this is quite a long process. So we're now at almost the last iteration. And what you can see on this progress bar are a few things. So we see the iteration number, the silhouette of the previously found iteration's pulse train, and the covariance value. And then next to these values, we can keep track of the progress by the progress bar. Then after doing this, we will finally do a check on the duplicates. And once those have been removed, we get the results on the screen, which you can see right now. So if we take a look at these results, we can see that we have a total of 8 different motor units that have been identified. And from these 8 different motor units, we first need to go towards the editing phase to see if we can get any artifacts distinguished, as Julien has already told in his part of the webinar. So to do so, we will move over to the addition pane on iSpin, and we start by importing the previous data set, and then you can see motor unit by motor unit the instantaneous discharge rate on the top and the actual spike train on the bottom. So we now first go by them one by one quickly to get an idea of what we need to do. And for one motor unit, we will actually show you the steps you could do in the editing phase. Because if we look at this specific motor unit, you can already see that there's some outliers in the discharge rate in the plateau phase, and we're gonna be correcting for some of those. And you can actually do so by using the Add Spikes button that I'm moving over to now. And then as soon as you've clicked this, you can get this draggable box, which you can select spikes to add. So you can already see two higher amplitude spikes that are not marked by a circle. So let me mark them just for a short while. And actually we want to remove those specific, or actually add those specific spikes to the total set. And then you can see what that does to the discharge rate. So the two outliers there have been removed. And then finally, it's important to reevaluate the window again so that the algorithm is recalibrated there. And if you're, what you can do is after doing this is check if the silhouette value that you can see here in the top right will be changing accordingly. And actually, we should expect that it increases. So let's quickly check that. Well, you do see a change in value. It's very limited, but you then can get an idea of what you can do on this editing phase. Well, after doing this for all the motor units, we are not going into that for the sake of time. We can also make a plot of the different spike trains with respect to the force profile. So we're showing that in the plot currently coming up. And in this case, you can see the unsorted version of all the spike trains that have been identified. And what's always nice to do at a later stage is to sort these, and you can actually see them getting orderly recruited. But it's a kind of a nice summarizing step to end with after the editing phase. Then we're gonna move over to the live decomposition phase. And this live decomposition phase again starts with making some settings. The settings that we have are the target for the MVC, which should at least be similar to what we did previously, and actually lower may still be okay. Higher will likely result in spikes that are have not been trained on. Then there's also settings on the ramp and the plateau phase and the amount of iterations. So once again, we will get this preview of the force profile. And then in this window, we're going to see the actual discharge times in a bit. And this window on the right side is the recorded force. And here, these two horizontal bars are used to give us feedback on the force production. So once I start the video again, then you will see that these different bars will start moving up, and the subject should contain the circle here in the bottom within those ranges. So let's get started on that part. So again, we'll make the additions to the training profile that we want to use, and then start the actual plot so you can see that it updates on what we're going to do. And once again, you can see our subject sitting on the foot pedal, overshooting already a bit in the rise time of the force production. And then here, just for the sake of illustration, we also let the subject overshoot this force profile a bit so they can see multiple motor units coming active at this time. And of course, when we drop the force, less motor units are needed to realize this force. So you will see that not all motor units are firing at this specific instant. Then actually our subject can repeat doing this for a prolonged time and actually alter the rate of force production based on the feedback provided. And actually that brings me towards the end of this short demonstration. So as Julien said, iSpin Saga has been an adaptation from iSpin Live. And to learn more about this, please visit the link in the description. So thanks all for your attention, and I think that makes it time for questions.
Yes, correct. Thank you, Joost and Julien, for the nice presentation and also demonstration video. We're now open to any questions that you may have. Feel free to write them in the chat box. I see we have one here already from Dylan Carter. And he asks, have you done any testing using smaller muscles with less motor units, say in the upper limb? And for this, Joost, could you start with answering?
For this specific and using it with iSpin, I haven't done tests on the really small muscles. Maybe Julien has done this. I'm not sure if he can add to that.
So for the upper limb, actually, I'm working a lot on the biceps and tricep, but I don't think it's small muscle. But I know me, I haven't doing. I haven't did it, but I know that there is a lot of research that's using this small muscle, the first interosseous muscle, and I think there is a lot of work on this muscle, and I think it's also a good muscle to have a good motor unit, because as you can see in my presentation, the result is not the same if you change a muscle. So, some muscle are better to do motor unit decompositions than other, and this muscle is a good one. So for the as I know, yeah, you can at least test. Yeah.
Yeah, thank you, Julien, for answering the question. We have, and Dylan also for asking the question. We have another one in the chat from Yuan. A single muscle contains hundreds of or thousands of motor units, but typically only a few dozen motor units are separated using algorithms. How can this result be understood or explained?
So yes, so we have to know that the EMG decomposition is biased toward certain motor units. Typically, when you do EMG decomposition, the motor unit with the largest action potential in amplitude will be recognized first. And so there is several answers to this question. So first, you can increase the number of motor units recognized by increase the number of observation. So the more you put electrode on your muscle, the more you will have a motor unit. But indeed, if you in my presentation, my high density electrode was with 64 electrode, and so I have only 30 motor units. So first, yes, I am biased toward the largest motor unit, but also you have in total hundreds of thousands of motor units, but in the force amplitude of the task, so 20%, 40%, not all the motor units are active. So you can not see the motor units that are not active because they are not producing action potential. So you have to reduce the number of motor units to the number of motor units that are indeed active. And then, yes, you are biased also toward the motor unit that is close to our high density surface EMG, because typically the recognized, the identified pattern of motor units is motor units that are not below 2 centimeters below the high density EMG. We are not getting motor units that are deep into the muscle. So if you want motor units deep into the muscle, you also can use intramuscular high density EMG. And finally, my last answer to this question is when you have some motor units, not all, obviously, you can also try to use some algorithm to predict the full population behavior. Because as I mentioned in the beginning of my talk, some motor units are very flexible between two motor units, but also some motor units have the same behavior. And so you can model the full population of motor units by using only small percentage of motor unit recorded. And so you can simulate the population of more motor units within your muscle targeted. I hope I answer your question.
Yeah, thanks, Julien. And also Ying Yang for your question. We will move on to the next question from Wanboom, asking, is it possible to load already saved EMG signals and then perform a decomposition?
iSpin has been specifically developed to actually record the data in parallel as well. So if I'm not mistaken, that's not that straightforward. As Julien already mentioned, iSpin has been developed together with the group of Simon Avrillon, and they created another open source toolbox named MuEdit that actually shares kind of the same graphical interface. And that could be a first step to have a look at already recorded data. And actually at TMSi, we're also working on creating a interface to do this based on data recorded with data from, for instance, Saga. So that's also something that you can keep a lookout for.
Yeah, if I may add something, indeed, Simon Avrillon has developed also an application which is motor unit edit, where you can import EMG signal to decompose it. And in iSpin Saga, you also have a button to import separation vector and classifier from previous experiment. And as it is developed by the same team, it is highly compatible. So you can do offline decomposition from some previous EMG recording, get some separation vector, and then try to apply it to have some real-time feedback. However, just want to award you that this separation vector and classifier works for a very specific localization of your identity EMG grid, surface grid. And so if you do it, if you try to decompose offline EMG signal and you try to use this separation vector on a real-time feedback, it will works if you are not moving, if you are not removing or moving the surface EMG, but probably it will fail if you did move your grid.
Thank you, Julien and Joost, for answering the question from Wanboom. We will move on to Boudewijn. He asked, thank you for the presentation and demo. When dozens of motor units are active, how labor intensive is the manual editing? How much time does this post-processing step take relative to the experimental duration?
So it's a great question. So the manual editing first, I will say that it's highly reliable across operator. We already published a paper on this. And so it's a paper of François Hugues and collaborator in 2021 in the Journal of Electromyography and Kinesiology. And so how many time it takes, I would say it depends on your results. But basically, I would say for 20 motor units, you can have between 5 and 10 minutes of editing. More 5, I would say. Basically, for the real-time feedback, you don't want bad motor units. I would say bad motor units that poorly defined motor unit. So if it is too hard to edit, it's probably not a good motor unit to use for real-time identification. Because if it is badly defined, with time, with fatigue, with a lot of things, the separation vector and the classifier will be poorly fit the motor unit tracking, and so you will lose basically your information. So I would say that it's quite quick, because you want to keep only the good motor unit, the best, I would say. This is also why we put such a high threshold for the silhouette value, because we only want the best motor unit. I hope I answer your question.
Thank you, Julien, and also Badoline for the question. I will move on to the next one from Alice, asking for the biceps brachii, how is the results of decomposition? Are you able to detect several motor units at a different level of contraction? And a second question, have you tested if there is a relation between the electrolocation and the quality of decomposition? There is a relation with the location from the innervation zone.
So there is a lot of question here. So first, yes, with the bicep brachii, I have some good motor units. Around 20, I will say, with 64 electrode, not 32. With the more electrode you have, the better it is for decomposition. And I speak with, I'm using the Saga from TMSi data, obviously. And so yes, I have some motor units. The idea is you want to put the grid on the belly of the muscle. The best is to align the column or the row of your high density electrode with the muscle fiber, because if you align it, you will have more recording of your action potential as it goes through the muscle fiber. The fact is that with biceps, it's bipenate, so it's hard to align with your muscle fiber. This is why it's better. You have better result with the tibialis anterior, where it's really simple to align your matrix with the muscle fiber. So yeah, you have some good results, and I'm not sure I have answered all these questions, because I don't remember all the question. Can you read it again, Colette, please?
Yes, sure. So the first part of the biceps brachii you answered, and the second part, have you tested if there's a relation between the electrolocation and the quality of decomposition? And there's a relation with the location of the innervation zone.
So I don't know for the innervation zone. I will say that the idea of EMG signal decomposition is to have a unique pattern in the space representation of each motor unit. So it's more the muscle fiber and the action potential going through the muscle fiber that will be important. So I'm not sure I can answer your question, but when I'm doing EMG decomposition, I try to position the matrices on the muscle belly, the most in the center, and also I try to think about the orientation of the muscle fiber more than the innervation zone. So yeah.
Thank you, Julien, and thank you also everyone for the questions. We have 1, 2, 3, 4 more in the comments that I will read. And if there are any more, please send us an email, because we are now getting towards the end of the time and I want to make sure we can answer this. But any further questions, we're happy to give answers through email and have a one-on-one conversation with you as well. So please do not hesitate to email us afterwards too. I will move on to the next question from Dylan. Is it possible for this program to decompose two muscles at once, possibly tracking synchronous firings across different muscles in real time?
So yeah,
I can take that question.
okay,
Yeah.
go
ahead.
go ahead. No, go ahead. Go
Yeah.
ahead.
Yes, this is indeed possible because in the screen that I showed at the start of the video, we configured which muscles to record from. And actually there's two fields there which you can fill out which muscle you're recording from. And if you would use two 32 channel grids, you can then use two muscles and track them simultaneously.
Nice, thank you, Joost. And Dylan, again, for your question, we will move to yin yang. Is the blind source separation algorithm based on the assumption that each source signal is independent? If so, do the decomposed motor unit signals still have any correlation, such as synchronization characteristics?
So no, the blind decomposition suppose not that the sources are independent, but more that the sources have a unique spatial distribution pattern. This is why if two motor units share the same space, the model will put in the same motor unit discharge pattern. So if you do EMG decomposition, you can still try to try to look at synergy of motor neuron. You can still look at correlation between motor neurons. So some motor neurons will be highly correlated. Some motor unit will not be highly correlated. Really, the key point is that two motor units must have a unique spatial distribution pattern. And if not, these two motor units will be merged in one by the model. I hope I answer your question.
Thanks, Julien, and also Yin Yang for the question. We will move on to Fungia and the question of MU edit seems need add specific Saga EMG channel map before decomposition instead of loading poly5 or MATS file straightforward.
I can take this question. You're correct. As Julien explained, MU-Edit has been developed in parallel to iSpin, and they align the data structures there. So we've actually worked on creating a interface to load existing files that are recorded with a TMSi device into this infrastructure. So if you want to start using that, please be in touch with us and contact us after this webinar so that we can see if we can share the resources to do so.
Nice, thanks Joost and Fanji for the question. We move now to Raphael, asking thanks for the presentation. To which extent would you say that separation vectors and SILP and R thresholds may be applicable in tasks involving changes over time in MU action potential waveforms, example fatigue? To ask it differently, how do these separation vectors relate to the action potential waveforms?
So yeah, as I said, it's rely on the spatial distribution of your motor units. So yeah, it's rely on the form of your of your action potential. So for sure, for example, if you have some fatigue, that will change the shape of your action potential. The separation vector will become more and more outdated, I would say. So there is several possibility to answer this question. First, the first thing that you can do is for each contraction, for each task you did, for each trial, you can update the separation vector so you can follow the change. And so your motor unit, as it will be the separation vector and the classifier will be updated for each trial, then you can still track the motor unit and do some real-time task. Also, these motor units that you choose to do real-time tasks are, as I said, the best motor units. So the change maybe will bring more and more error, but you will not lose totally your motor unit. And so I don't know what is the task you plan, but if you do some correlation or coherence analysis, yes, you need a perfectly fine discharge pattern. But if you only do some visual feedback of the discharge rate will be still more or less accurate. So it's a compromise. But yeah, you can also try to update the separation vector and the classifier after each trial to follow the modification of the shape of the action potential.
Thanks, Julien. We have one more final question in the chat before I move to the closing slides, and that's from Yin Yuang. Can motor unit decomposition be performed by segmenting the signal during a signal-sustained submaximal isometric contraction? Are the motor unit signals comparable between different phases?
So I'm not sure I understood your question. Is your question depend on the amplitude of forces, like at when you do a ramp at 20, 30, 40%? So if it is the question, for sure, you have different, you don't have different motor units, but you recruit more and more motor units. And by recruiting more and more motor units, you're recruiting the largest one. And so if you do decomposition at 10% of MVC, you will have a result, some motor unit. If you do at 20% of motor unit, it's you will probably have other motor units. And so by doing so, in fact, you can build what we can call a library of motor units. You can do a contraction at 10, 20, 30, 40, and try to decompose for each amplitude, and then merge into a huge matrix all the separation vector. And so some motor units will not be able to be tracked at 10. The motor unit you find at 10 will not be able to be tracked at 40 because there is some new motor unit that share the same space, but not all motor units though. You saw it in my presentation. And so you will be able, by doing so, to build like a good library of all the motor unit, the smallest one because you identify it with small contraction, and the biggest one. And so yes, indeed, you will not have the same motor unit if you change your force production. I hope it answered the question.
Thank you, Julien, and thank you everyone for your very good questions and nice discussion. I will move on to some closing slides now. So maybe you have a question of where can you access and download iSpin Live? And on our website, if you go to tmsi.com and then under menu, you will find our new integrations page. And there you can see a whole list of integrations. And then click iSpin Saga Live to get more information about it. And lastly, to also download the data. Would you like to also see a live real-time motor unit decomposition demonstration in person? Then you can also visit us in ISEC if you are there. We will be there in Japan from June 26 to 29. There you can also see our HD, EMG, and EEG products. You can also have consultations with us for some personalized advice. So feel free to come chat with us at our booth or schedule a meeting as well with us. We'd love to see you there. Next, this meeting, of course, this webinar was really focusing on iSpin Saga software. But if you want to learn more about the textile grids, you can visit our webpage and also look at our old webinar where we've really focused on the grids and the science behind that. I will also be putting a link to this in the email that I send out after the webinar. And lastly, we want to thank you all for joining The webinar today. So we have a webinar offer of a HD EMG starter package where you will receive 50 grids and 200 adhesives for free with the TMSi HD EMG full solution package. So feel free to reach out to us if you are interested in this offer. Lastly, I want to thank you all for joining and also thank Julien and Joost for the presentation and also the demonstration. And I wish you all a very nice rest of the day.